be talking about this review paper titled as Cell-Type Deconvolution Methods for Spatial Transcriptomics. This was published recently in July 2025. So I'll be talking about this paper today. So spatial transcriptomics measures the gene expression while preserving the spatial arrangements of the cell. There are two technologies for the spatial transcriptomics, imaging-based and the sequencing-based, where imaging-based looks at the single cell or subcellular resolution, but this is limited to around like 200 to 1,000 genes. While sequencing-based spatial transcriptomics technique looks at the whole transcriptome, but the thing is, with sequencing-based transcriptomics, each spot contains multiple cells. That is, like, it lacks the single cell resolution. So, like, what is spot deconvolution? Like, it's a computational method to estimate which cell types are in each spot and what is the proportion of each cell type. So why it matters? It resolves the cellular composition beyond the spot level resolution. It maps the cell type distribution, looks at the spatial density and spatial relationships. It also helps to look at the discovery of tissue changes in development, aging, and diseases. So, like, at each spot, like in this figure, we can see each spot contains like different cell types. And then from spot deconvolution, what we want to do is to estimate the proportion of these cell types in each spot. So what this particular paper that today we are talking about, it explains about the inputs and outputs for deconvolution. It does categorize the methods based, whether it is based on reference or it does not use a reference, it is reference free. It tries to talks about the integration of single cell RNA-seq data, spatial coordinates, and histological images. Moreover, this particular study, this paper, review-based paper, they have made a web-based interactive table where we can explore, filter, and compare different types of tools. So on the right-hand side, I'm showing the image, just the image of this particular tool, where they have different filtering types based, and we can take a look at that, where they compare different tools. So in the very first figure, they talk about the general principle for cell-type deconvolution for spatial transcriptomics. Also, to mention this, in this particular paper, they are particularly talking about only sequencing-based spatial transcriptomics. So, like in the next slides where I say spatial transcriptomics, from the paper, I'm referring to the sequencing-based information. So that is what we are interested in. So in the very first inset, in the principal workflow, we collect the sample for the spatial transcriptomics analysis, and then do the data processing and like try to generate the counts metrics. And we also look at we try to annotate the different cell types, annotate the single cell RNA sequencing data, and then try to extract the features with different modalities. So in this figure, I'm showing like the genes and the cell types for different types of cell types, as you can see here, and then the signature metrics, reference signature metrics that they're looking at. So for spatial deconvolution, many of the methods, they depend on something called as pseudospot generation, which I will talk about. The paper also mentions a separate section for pseudospot generation. And then for the spot deconvolution, we can also look at the segmentation and extract features from histological images, getting the segmented nuclei, how many nuclei are there in each spot. We can try to get the information from this, and then combining all this information, like using this as an input for starting the process of deconvolution, applying different strategies, the possible type of outputs that we get from the deconvolution is related to, like, we can get like a mapping metrics here, like the cell versus the mapping probabilities, and also get the cell locations, like in each spot, these tiny little colors that you see, these represent the different proportions of the cell types. That is in this C middle figure. Panel C is deconvolution matrix. where for each special capture unit, we can get the cell type proportions, the cell type counts. And then based on like this information, we can reconstruct single cell gene expression metrics, like the genes versus gene expression. And we can look at the different expression values. And then this deconvolution outputs we can also use for downstream analysis and try to visualize cell type distribution, looking at the different cell proportions, detection. We can also detect cellular microenvironments. And then another, like what we can try to look at is cell to cell interactions. We can try to integrate with other analysis tools for this special transcriptomics and try to look at interactions between the cells and also look at which cells have more affinity to be the neighbors or the interactions between them. For pseudo, as I mentioned in the principal workflow about the pseudospot generation, most of the methods for deconvolution, so the pseudospots are simulated from the single transcriptomic spots, from the single cell data, and these are then used to train the models and benchmark the deconvolution method. But these also, the pseudo spot generation, these based on the sampling strategies could be random or guided, and then they help to address the scarcity or the imbalance of the data, especially trying to find rare cell types. But this pseudospot generation method does have a limitation that it does not fully capture the spatial complexity, or it does not take into account the batch effects. So we as users, we need to keep this thing in mind, and the quality of the spot deconvolution results, it depends on our single cell annotation results. So these, like when we are trying to do the spot deconvolution method, our inputs should already be processed and QC'd to consider the batch effects and these things. So this is one important thing that the deconvolution methods don't address. So we should keep this in mind. Then a good thing about this review paper, what I liked is they have tried to classify all the cell type deconvolution methods for spatial transcriptomics. And in this review, they have looked at all the papers from PubMed and bioRxiv till March 2024. And also they have included the papers which were peer reviewed from March 24 to December 24. So in this classification image for the deconvolution methods, we can see there are like negative binomial distribution methods. So the most famous one is the cell-to-cell location that has been quite benchmarked in different studies. And also this paper, when they tried to compare all these methods, cell-to location has been one of the most, like the method that stands out. Then there is this another framework called as the Bayesian framework. So this involves RCTD, mass decon, assigned spatial scope. So comparing all these methods, RCTD stands out compared to all these methods which are based on Poisson or Poisson gamma distribution framework. And then they also they have also classified these methods such as the regression framework, nonlinear, optimal. And then also they have these deep learning methods and other deep learning architectures that they look at. So some of these, such as the Let's Mix method, this kind of uses the special DLPFC method for their study. So that was interesting to know that the special DLPFC study was used for making this Let's Mix method. And then another one also called as the metrics factorization. This particular spice mix also used the special DLPFC dataset for their method. And amongst all these methods, two methods, like after classifying the two methods that like they have talked about the most, which kind of stood out the most, was cell to location and RCTD. They have also, in the figure 3, they have also characterized these different methods for deconvolution. So in this figure, they are trying to show the inputs for that go for the deconvolution method, what type of programming language those methods are based on, what type of outputs we get. Do we get proportion counts or the probabilities? Do we get the mapping or cell location or single cell gene expression? Then also, as from the previous figure 2, what type of framework these methods are based on. And they have also looked at some previous benchmark studies, and which of the methods have highlighted those methods is what they are trying to compare in this. So all these methods, like what input goes into these methods, is it like the metrics, the image data, or the coordinates that go as the input? And then they have also classified these methods as reference free and reference based, some which require images and some which require coordinates. So the one in this, like the cell to location requires the image, but like this is also based on the Python programming framework, while the CTD is under the reference based, but it does not require image data. And also this CTD is giving us the output as proportions, and the programming language it is in is, so that is most compatible with the type of data that we are trying to work on. So like the different colors over here, like on the left hand side, represent method groupings. Like and also the colors you can see if it's like does it require like the reference or no, like the reference is optional, does it require the coordinates or the images. So these colors you can see can tell us, like if it's the dark color, it requires the reference, it requires the coordinates and the image, and half, it shows that it is optional, or it might need the image data. So this is like this was a good like table which kind of summarizes all the different types of methodologies and the frameworks all together in this particular image. Then again, in the fourth figure, they talk about multimodal integration for spatial transcriptomics and special multimodal integration for special transcript deconvolution. So like all this information, like looking at the single cell data, where we have like the genes and the cells metrics, where we annotate the cells, annotate the single cell RNA sequencing data, and try to do the gene selection process, looking at HVGs or differential expression genes, and also try to do the special mapping, and then trying to apply some sampling strategies to get the pseudo spot counts. It could be random or multimodal distribution or MCMC, Markov chain, Monte Carlo strategy, to get the pseudo spots and aggregate those counts, and then try to connect the cell types, try to get the information about cell type specific gene signatures and special transcriptomics data with the pseudo counts. And also these methods, it would be good to see integrating these methods, the image data, in addition to these, because it does not, like these sequencing based deconvolution methods, they do not have like a standard framework where it includes the image data. So, and also like it is a challenge to have all these information, try to connect all these information, and then like with the single cell data, and then for special transcriptomics, we need the coordinate data where we want the special locations, and then we can look at the different graph structures where we, in the special transcriptomics, we do the pre-processing, like do the normalization steps, dimensionality reduction, and we also try to extract different features, look at the special normalization results, we look at the gene expression from those. And so yeah, like these different modalities, so it can be good to have some future work where they talk about the integrating all these information for special transcriptomics. they like with all this information, they have like what are the key questions that can guide us to do the method selection. So in this paper, they have these set of key questions to guide method selections, like looking at the tissue characteristics, if it's like structured or disordered, or whether we how much previous knowledge is available, where we want to where we want to use the reference, we want it to be reference free, and then also the type of input data sets that are available for given data set that we are interested in, whether we have single cell RNA-seq information, whether we have information of image data or the special coordinates. And also another question that can help select the method is looking at the proportions, the deconvolved expression, or we want to look at the single cell maps. And then like how was this particular method validated and benchmark is also one of the questions. So with all these questions in mind, they have created the web-based page that we will look at to select the type of method that is suitable for our given data set. Also, like these deconvolution outputs we get as proportions, a type of compositional data. And this data, like it could be, the proportions sum to one, but the challenge is that how these, like the composition, and then how, whether those composition summation and what type of special statistical treatment it might require. So some methods might misinterpret this relation because of the sum constant. And this is one of the paper suggests about doing the compositional data analysis to use log transformations, log ratio transformations to analyze the proportions properly, and so that it can enable to get accurate modeling results and the interpretation for the cell type abundance in the given data set. And these, like, they talk about the different perspectives that can be considered for development to develop the reference-free deconvolution methods for rare uncharacterized tissues, and also method to enable the multi-sample or like the comparative analysis, and to improve multimodal integration, especially where we have the image and the spatial data, and more benchmarking with the diverse data sets. And with a VZIM HD coming up, advances in high resolution spatial transcriptomic data, it might require new deconvolution methods. We can take a look at this table, this website that they have, where they look at the different deconvolution methods. So in this, they have all these different filters, such as does the annotated single cell RNA sequencing, whether it is reference based or reference free, both. And then like the type of algorithm that you want to look at, whether you want to look at regression type, Bayesian framework, or dimensionality reduction, or consensus algorithm. Say, so in the reference free with using all the types of algorithm, we can see the type of table that we get is the method name, the programming language it uses, whether it requires transcriptomics, special transcriptomics coordinates, and also information about whether it is reference based or reference free. So if it is both reference based and reference free, it gives us the information that it is both. And then whether it requires image, yes or no, and the GitHub link to all these pages. So you can also select how many entries you want to see at once. So this also involves ST deconvolve, transformer ST. So this way we can select which type of method we want to look at, and then whether we want to just stick to, in that case, it kind of gives us this information. Some which also have programming framework in Python. And so RCTD, as we can see, is based on R, and the cell to location uses Python. So if we look at all the programming languages, they have total 67 entries. They looked at 67 different studies in this particular field. And then they also have information with like what type of columns you want to display along with the method name, the publication date. You can also select it to the journal, and then other counts, whether you want the special transcriptomic counts, you want to look at the coordinates, and then about the image and the codes. And also they have also included the tutorials. So the studies, the deconvolution methods that have different tutorials linked with those deconvolution methods. So they have made an attempt to integrate all this information together. So I feel like is a good overall review and a summary to look at the different benchmark methods. They also have in their supplementary tables, they also have this table where they have these four supplementary tables where they talk about the existing tools for deconvolution. And then again, it's the same method that I just showed on the web page, but they have these supplementary tables already about the different methods, whether it requires coordinates, images, outputs, and what type of program it is included in, whether it is classified as reference based. Are these methods reference free? Is it involving both reference free and reference based methods? Then second table again includes all this information, like global description of each cell type deconvolution methods, where they have extensively added the method name, publication date, and all the article informations, article authors, URL, and like this is the background for, I think, the web page that they have developed. And another supplementary table, table 3, is the description of each key characteristic of deconvolution method. So the method description they have here is like a general description, but whether it requires the special counts or not, they have just included these terms, whether it's the Bayesian regression, what these terms mean, so that it is easy to look at the data. And in table 4 is the common abbreviation. So this is like a table of different terms, like what are the different abbreviations commonly used in the deconvolution articles, whether like NMF stands for non-negative matrix factorization, and all this information. CCA is like canonical correlation, KNN is K-means neighbors. So like for different analysis terms, a brief like one line description and definition they have mentioned in these supplementary tables. So yeah, that is what this overall paper is about. And yeah, I liked the way they have summarized all the different benchmark studies until like December 2024, peer reviewed until December 2024. And it's an extensive overview that they have. And they also, in the paper, they also have a form where they have given the option if you want them to add some methods which are not included in this particular paper, they have these Google form where they ask you to like submit those, and they will try to add that particular method in their toolbox, the deconvolution method toolbox. So yes, this is what I had for this particular paper. Thank you.